So, good morning. For today's journal club, my topic is high resolution spatial transcriptomics and cell lineage analysis reveals a spatiotemporal cell fate determination during craniofacial development. This article was published in Nature Communications with impact factor of 15.7 on 12 May 2025. So, following our terminologies which we need to understand, there is cranial neural crest cells. They are multipotent progenitor cells that contribute to craniofacial structures growth. And palatogenesis, it is development and process of formation of hard palate. Then mesenchymal progenitors, they are precursor cells that will give rise to connective tissues like bone, cartilage and fibroblasts. The sequential fluorescence in situ hybridization, this fish, it is high resolution spatial transcriptomics method which is used to map gene expression in tissues. Then there is scRNA sequencing, single cell RNA sequencing. It is a technique used to profile transcriptomics at a single cell resolution. Then there is lineage tracing. It is an experimental technique to track progenitor cells and their differentiated descendants. Then odontogenic lineage, it is a cell giving rise to tooth and supporting structures. There are then perizymal cells, they are mesenchymal cells surrounding developing muscles, crucial in palatal morphogenesis. There is osteogenic lineages, they are progenitors differentiating into the bone forming cells. And then there is spatial transcriptomics. This integrates gene expression with anatomical location for studying dynamic developmental process. So in this article, they developed a first high resolution spatiotemporal atlas of craniofacial mesenchymal development and demonstrated that the cell fate diversification occurs earlier than the appearance of anatomical structures. They combined sequential fish and scRNA sequence and lineage tracing, providing unprecedented insights into the craniofacial odontogenesis. So they hypothesized that the cranial neural crest derived mesenchymal cells undergo early fate diversification and can be tracked using lineage specific markers before palatal shelf formation. So the aim was to map the molecular and spatial landscape for craniofacial mesenchymal development, and the objectives were to identify lineage specific progenitor population during palatogenesis, trace their spatial and temporal dynamics using sequence fish, and validate bioinformatics predictions with in vivo lineage tracing. Also, and establish a dynamic resource for studying craniofacial anomalies. So the methodology, it was an animal study. They approved, they had the ethical approval. They used the mouse species C57BL6J, and they used transgenic lines, that is SOX9, to trace SOX9 mesenchymal progenitors, to trace odontogenic lineage progenitors, and to trace perimysial lineage progenitors. And they used tamoxifene induction, which labels the early progenitors and track their descendants in the late stages. The tissue collection and processing was done at these days of the week, and fixation was done with decalcification, and embedding was done. They did cryosectioning to see the tissue palatogenesis development. The single cell RNA sequencing was done to dissociation of palatal tissues into the single cell suspension. The library preparation and sequencing using 10x genomics chromium was done, and they counted around 80,000 cells across 5 stages, like in embryonic 12.5, 13.5, 14.5, 15.5 and 18.5 of days of the development of mouse embryo. And pipeline analysis was done where they integrate Surat integration was done to combine the data across stages. Differential expression analysis was done to assign cluster identities, and marker genes was done to identify osteogenic, odontogenic, perimycial and midline populations of cells. The spatial transcriptomics was done, sequential fluorescence in situ hybridization to map the gene expression directly in the tissue section, and a custom panel of 94 marker genes was selected. The known lineages of the markers were these genes were selected, and newly predicted markers from SC single cell RNA sequencing clusters were also identified. The tissue sections from these weeks were imaged, and they used Scanopy and tissue maps to identify to analyze, visualize the lineage distribution in 3D spatiotemporal context. Then lineage tracing experiments were done. They used pre-LOX-P system to tamoxifen inducible pre-recombinase, where they used COX-9 3ER, TFAP2B 3ER and HIC1 3ER cross-bit TD tomato reporter mice, and the injection were labeled in 9.5 and 11.5 weeks. They injected with tamoxifen label, and the embryo harvested later in this week to check whether the labeled cells contribute to osteogenic, odontogenic or perimycial lineages, and the detection was done by immunofluorescence microscopy and RNA-scope in situ hybridization. They validated the experiments by RNA-scope and immunofluorescence. The statistical analysis, they did filtering. The cells were filtered by gene counts, transcript counts and mitochondrial read percentage. The integration of SCVI tools for batch correction and integration across replicate stages, and the clustering was done. Lead-in clustering resolution 4.0 was done to define distinctive cell clusters, and differential expression they used to identify stage-specific and lineage-specific markers. They visualized it using UMAP plots, dot plots, and integrated spatial maps across E12 to E15, E12.5 to E15.5. The results were as follows. They identified distinct mesenchymal population which was either osteogenic, odontogenic, perimycial or midline lineages. The early progenitors heterogeneity was observed at the embryonic week 12, day 12.5 before the palatal cells were formed, and the TFAP2B and HIC1 progenitors contributed to the odontogenic and perimycial lineages respectively. The spatiotemporal migration of osteogenic and midline cells were mapped across the palatal development. Coming to discussions, so the CNCC, that is cranial neural crest cells, derived progenitors diversify earlier than expected, and the spatial transcriptomics enabled tracking of cell-to-cell interaction and dynamic lineages transitions. It also suggests that conserved regulatory mechanism guides the late stage of CNN migration, CNCC migration and differentiation. So the strength of this study was it was the first dynamic multi-stage craniofacial mesenchymal atlas It integrated SCR, single cell RNA sequencing, sequence fish, and lineage tracing, increasing robustness. And the data was publicly available on face-based for community use. The limitations of this study was they primarily focused on palatal regions, and other craniofacial regions are needed to be explored. The sequential fish is limited by panel size of detectable genes, so they can use different genes, and lack of functional validation of all identified progenitor markers was there. So the study design was animal model and plus multi-omics profiling and lineage tracing validation, which so it compliances with ARRIVE guidelines. So this article also follows the Nature Communications guidelines. So in the ARRIVE guidelines, they did not mention about the sample size, which was not explicitly calculated, and the inclusion-exclusion criteria was not clearly stated, as well as there was no mention for randomization, likely because there was no randomization. The rest other guidelines were followed by this article. So this is the general term.